Hello everyone, this video is about the Calvin cycle. The content's made from grade 12 biology course SBI 4U from the photosynthesis segment of the metabolic processes unit. So some important information we need to know is that the Calvin cycle is a light-independent reaction which requires no light to work, occurs in the stroma of the chloroplast as seen in the bottom image. The stroma is the fluid-filled section of a chloroplast cell, comparative to the cytosol in animal cells. The purpose is to convert carbon dioxide into glyceraldehyde 3-phosphate. G3P is used to make glucose molecules which are used for energy in plant cells, or converted into cellulose. We need 6 full cycles to take place to produce 1 molecule of glucose. The Calvin cycle has 3 phases. Phase 1 is the carbon fixation phase. Phase 2 is the reduction phase, and the third phase is the regeneration of RUBP. This will make more sense in the next slide. So, the Calvin cycle, as stated, occurs in the stroma of the chloroplast. So first we have our carbon fixation phase, where we're going to take the example of 6 molecules, as this would make 1 glucose molecule at the end, but this occurs 1 molecule at a time. So we have 6 carbon dioxide molecules, which is a 1 carbon molecule, joined together with 6 ribulose 1,5-bisphosphate molecules, which is a 5 carbon molecule. What makes this possible is the enzyme Rubisco, which is very important. So the product is 12 3-phosphoglycerate molecules, which is a 3 carbon molecule. In the next step, we have 12 ATP, which are converted into 12 ADP and 12 phosphate groups, which are then added to make 1,3-bisphosphoglycerate. So the phosphates get added to the first carbon and make 1,3-bisphosphoglycerate. This step on is known as the reduction phase, which is phase 2. And next we have 1,3-bisphosphoglycerate turn into 12 glyceraldehyde 3-phosphate molecules, which is another 3 carbon molecule. Now here we have NADPH made in light-dependent reactions being oxidized to make 12 NADP molecules, NADP+, which reduces the 1,3-bisphosphoglycerate into 12 G3P molecules. In this step, we also have 12 phosphate groups leave 1,3-bisphosphoglycerate. So from there, we have 2 G3P molecules leave to make a glucose molecule, which is a 6 carbon molecule, and other sugars. Then the remaining 10 G3P molecules go on to be regenerated back to RUBP, or ribulose 1,5-bisphosphate. So in this step, we have 4 phosphate groups leave, and we have another 6 ATP molecules being converted into 6 ADP plus a phosphate group, or 6 phosphate groups. So what this does is it allows RUBP to be regenerated, making 6 RUBP molecules, which allows the cycle to continue. Okay everyone, thank you for watching the video. I hope you learned something new. Good luck on your tests and quizzes. Thank you very much.